Assalamu alaikum, dear student. Today in this lecture we will discuss about the antigen, epitope and heptene. So first of all, what is antigen? Actually, antigen is defined as any substance that satisfy two distinct immunological properties. The first one is immunogenicity. What is immunogenicity? Ability of antigen to induce immune response in the body, both humoral immune response and cell mediated immune response. Humoral response mean the production of antibody, and cell response mean the involvement of the T cell and the conversion of the T cell to memory cell without the production of antibody. The second is antigenicity or immunological activity. Immunogenicity is the ability of antigen to combine spatially with the final product of the B cells or T cell, mean that antibodies and T cell receptor. All molecules having immunogenicity property also show antigenicity, but the reverse is not true. For example, heptene, which are antigenic but not immunogenic. What is epitope? Equally, antigen have a specialized area that's capable of sensitizing T cell and B cell and involved in the production of immunity or the sensitization of the T cell. And what is paratope? Paratope is the specific site of antibody that react with the corresponding epitope of antigen. It's called paratope. Epitope may be grouped into two types: sequential or linear epitope, conformational or non-sequential epitope. Sequential epitope actually present is a single linear sequence of few amino acids, and conformational actually are found on the flexible region of complex antigen having tertiary structure. In general, T cell recognize sequential epitopes while B cell bind to the conformational epitope. Now what is heptene? Heptene are low molecular weight molecule that lack immunogenicity but retain antigenicity. It means that cannot induce immune response, but what they do bind to the specific antibody or T cell receptor. However, heptene can become immunogenic when combined with larger protein molecule called carrier. It is observed that heptene carrier conjugate introduce antibody specific or epitope of the heptene, unaltered epitope on the carrier protein, and new epitope formed by combined parts both of the heptene and carrier protein. Now, heptene we classified as a complex or simple. What is complex heptene? Actually, it contains two or more epitope that can react with specific antibody, and the heptene antibody complex be visualized by various methods such as precipitation reaction. Simple heptene are contain only one epitope. Such heptene can combine to the antibody, but the heptene antibody complex cannot be visualized as it is believed that precipitation to happen. It required the antigen to have at least two or more epitope.